I wish you a good day, and I thank you for inviting me to share some thoughts on applying quality by design, which we will call QbD, in pharmaceutical dosage form development. In my talk, I will take a professional development stance, which I'll explain later what that is, to be a good practitioner in sharing knowledge. The knowledge I wish to share is how to integrate design and systems thinking to improve and develop continually. The objectives of this workshop are to provide an overview of the general principles and practices in the implementation of quality by design, with an emphasis on integrating design and systems thinking. 2. Illustrate the application of design of experiments in pharmaceutical product development and explore how this introduction improves understanding of quality by design. Elaborate on some advances in the application of design of experiments. From my perspective, in my talk, I will use a simple practical illustration of design of experiment, not the technical or statistical aspects, but simply the framework of a design of experiment to show how it improves our understanding of quality by design. Once I have done that, I'd like to emphasize a practical means to integrate design and systems thinking, and that would be an experiential learning process and a process of transferring knowledge. A work in progress, number 3, is how do I learn from experience? How do I design my thought experiments? Here I'm thinking about a nested N of 1 thought experiment. So I'll just introduce the notion of a thought experiment and designing a thought experiment. So the flow of this presentation is why design of experiments, how it improves understanding of quality by design, and what I can do with this information, or I will do with this information. There are a number of regulatory guidances and guidelines related to quality by design. Starting with the PAT guidance from FDA, a framework for innovative pharmaceutical development, manufacturing, and quality assurance in 2004, which I had the privilege to lead, and also ICH Q8, which I led until it's step 2. And this picture is from the step 2 discussion in Kyoto, Japan, November 2004. As the FDA lead for quality, I had the responsibility to sign off at the step 2 of that document. At step 2, it becomes draft and is made available for public comment. During those negotiations, I had insisted on 3 aspects: regulatory flexibility to leverage the efforts in the development, which called design space, to allow for flexibility, regulatory flexibility, and opportunities for continual improvement. Not to be one size fits all in our recommendations, which is the placeholder is called Procrustean. Procrustes is a character from Greek mythology who would invite guests to spend the night in his house with one condition: they had to fit exactly into his bed, like in the specifications we have in pharmaceuticals. If the person was too short, he would stretch them regardless of the pain and discomfort to fit the bed, or if they were too tall, he would chop off the legs. So he was a nasty person, a one-size-fits-all person, and a Procrustean is the name we give to that behavior. And epistemology was another aspect, theory of knowledge. How do I know what I know? Our education tends to be reductionist, analytical, divided into disciplines, and pharmaceutical development is a multidisciplinary field. And quality overall summary: quality itself are integrative. You have to put the pieces of the puzzle together and they should fit nicely so that the essence of quality is evident in the quality of the writing. So synthesis, in addition to analysis, is a key. So to be able to synthesize, how do I know what I know is an important consideration. So a lot of my thinking was influenced by Dr. Deming, and his theory of profound knowledge has 4 parts: theory of knowledge, knowledge of variation, appreciation for a system, and psychology of change. ICHQ8 brings in theory of knowledge. Knowledge of variation is covered in many different guidelines, and that is our left brain, the analytical part of the brain. The right part of the brain, which is creative and design oriented, is appreciation for system and psychology of change. And I want to focus on this part of the profound knowledge, theory of profound knowledge. In life, test teaches a lesson. Matters of mind, a double-edged sword, can facilitate and retard our development at the procrastination stage. What can I do to change that? I wish to share my experience in taking a development stance to share knowledge. I wish to distinguish between information and knowledge. Information interacted with, experienced in an application becomes knowledge. In our experiential learning, we mature, we develop and mature, and we are able to fill the gap between what we know and what we practice. So the list of guidances is ICHQ8, pharmaceutical development, Q9, risk management, which is now under revision process to reduce the subjectivity of risk assessment, hazard identification, and to address formalities related to review and risk-based decision making. ICHQ10 defines pharmaceutical quality system. Q11 is quality by design for drug substances. Q12 is where we bring in the notion of more flexibility for regulatory post-approval changes and continual improvement, to take a life cycle approach to product life cycle management. It is at step 5. It's not a final guidance. ICHQ 13 deals with the continuous manufacturing, and ICHQ 14 is a guideline on analytical procedure development, the QbD for analytical method, but it also goes back to revised Q2 analytical method validation. So quality by design of analytical method is paramount to getting started in quality by design of pharmaceutical dosage form. I believe Prakash is going to cover that topic after my talk. Pharmaceutical regulations, laws and regulations have been established since 1906, when pharmaceutical science and technology were not as advanced as they are today. And layers upon layers of amendments to the laws have established legacy practices which may not necessarily be science-based in the current thinking, or they're no longer current. So we have legacy products and legacy practices and layers of those, and there are many blind spots that many are not able to see how to make those practices current to be more scientific. So few blind alleys that some of us, the old timers, have spent time in, and the next generation needs to learn about that. So that is one of my motivation, is to be able to highlight some of these blind spots. With the elixir of formulation example that I will use, I'll go back and reflect on why did it take 70 years to open our eyes, because only in 2007 we established a test procedure for diethylene glycol. since 1937, if you review the epidemiological data from CDC and other places, you'll see tens of thousands of people have died because of the same mistakes, same error of using contaminated glycerin with diethylene glycol in our formulations. There are other blind spots, such as blindly following the market standard or not knowing how to use the market standard, which are the compendial standards. You have to be better than compendial standards, because when tested, any sample should comply with the compendial standards. But unless you aim higher during development, higher than the compendial standards, you will create situations where you have failures and the root cause may not be known. And one of the key gaps that exist is analytical method validation pertaining to physical attributes such as dissolution, particle size, and so forth. In solution chemistry, we have we can make a solution, make a standard curve because each part of the solution is the same. That assumption does not hold for physical attributes, because each physical sample is a different sample, and you need additional considerations to validate methodologies for physical attributes such as dissolution, particle size, and so forth. And we don't do that very well. My eyes opened to that when dissolution failure had become a national security issue. And one of the gaps that we explored and identified was the calibrator tablet that we use for dissolution can pose many challenges. And therefore, we have to pay attention to validity in terms of calibration, gauge reproducibility and repeatability of methodologies we use for physical attribute measurement. And I think collectively, because of the legacy blind spots, we have market standards as a QC test. Market standards are intended for a sample that a pharmacist or somebody independent can use to test the sample that they have, and they don't reflect the batch. So we have type 1, type 2 errors built in, and therefore we tend to be in a corrective action mode and not have continual improvement. And when we repeatedly get into deviations out of specification and root causes unknown, we blame the poor analyst and the cycle continues. That is a frustrating process, and in a highly competitive, cost-sensitive operations, that is or can be an incentive to rationalize and have deviant behavior such as bad eye or breaches in assurance of data integrity. The third topic close to my heart is to transfer my knowledge to the next generation. If I could send a message back in time, epistemology and a medicine maker. Going back to my alma mater, University of Cincinnati, in 2007 to receive the Outstanding Alumni Award, my thoughts for the message I wanted to leave behind was epistemology without contact with science becomes an empty schema. Science without epistemology is, insofar as it is thinkable at all, primitive and muddled. Albert Einstein. That's also the message I want to leave behind today, and I hope you will be able to receive it and make sense of it and understand it in your disciplinary context. So now let's flow with why, how, and what. Let's begin at the bottom. All models are wrong, some are useful. Please remember that. There are 4 types of systems: simple, complicated, complex, and chaotic systems. Simple systems, cause and effect relationships are self-evident. You don't need any specialized education and training to understand cause and effect relationships. Complicated systems, the cause and effect relationships are not self-evident without proper education and training. That's where pharmaceutical manufacturing is. Complex systems, cause and effect relationships are not known and need to be deciphered through research and development. And complex systems exhibit emergent properties, and emergencies can happen if complexity is not reduced. Chaotic systems exhibit extreme sensitivity to initial or starting conditions and are not predictable beyond average. Starting conditions can be us, people, materials, everything that we use to start our processing, including our prior knowledge. If anything is wrong, it has some significant impact. When we do a design of experiment as opposed to one factor at a time trial and error experiment, our intention is to understand the response surface, which is an equation such as this, and it can have a simple linear part, an interactive part, and multiple interactions. Although it is still a linear system, it's an approximation, a narrow approximation of something that can be nonlinear. Without taking a design of experiment perspective, we are blind to the interactions and the interrelationships between multifactorial problems. Formulations are multifactorial, and therefore design of experiments is a way to understand quality by design. Extending the notion of multifactorial complex problem, let's use a very familiar metaphor of 6 blind men and an elephant, which is a Western interpretation of an ancient Indian story, which misses the point of the whole idea of the story in India was to get deeper knowledge, which Rumi actually understood in the 1500s. And his poem is called An Elephant in the Dark. So you have two options: 6 blind men and an elephant, or an elephant in the dark. Which one will you choose determines whether you are a systems thinker or not. If I use the elephant and translate that to our organization, especially how our policies and methods are organized in different parts of the organization, different divisions, different compartments, product, method, process development policy, and if I use the process validation guideline 2011, stage 1, stage 2, and stage 3, and associated with that is bio and exhibit batch manufacturing, pre-validation activity and process qualification batches, and commercial manufacturing and continued process verification, that can be imagined as an elephant. Rumi's elephant in the dark provides a very simple strategy. We hold a candle in our hand and we walk in together to see the elephant. As multidisciplinary team members, we each hold a candle. And when we go together to see the elephant in the dark, which is the uncertainty, then we can see it. If we take the westernized interpretation of this tradition, Indian tradition, 6 blind men and the elephant, then we are not taking our blindfolds out. We are touching and feeling within the limits of our sensory access to that elephant. And even if everyone does that and collects their observations on paper, and then we try to overall synthesize the quality overall summary from that, will an elephant emerge to the reader? Mostly not. Therefore, elephant in the dark is a better strategy. We acknowledge the dark and we know how to remove the darkness with our candles, our enlightenment. On 19th October 2004, at the Advisory Committee for Pharmaceutical Science, I had outlined a critical path opportunity to think about how we can leverage ICH Q8, which is section P2, which is the development report. And the key there was to think about compartment modeling as we learn in pharmacokinetics, and to be able to link different sections such as 2.1.1, which is a section describing drug substance that we want to use in the formulation development, 2.2.1, so via pre-formulation studies and make sure that characterized pre-formulation work was done right, and so forth and so forth. So ICHQ8 quality by design was the basis to say, all right, key physicochemical biological characteristics of the drug substance that can influence the performance of drug product and its manufacturability should be identified and discussed in section 2.1.1. And we want to make sure the formulation development has utilized that in their design considerations and control strategy and so forth. Unfortunately, this is, I'm talking almost 20 years ago now, but much of the sector has not changed. I still see root cause unknown. Out of specification issues are more often built in R&D and CMC review, and they become the basis for common cause variants, where the manufacturing people have no choice but to say root cause unknown. We have cognitive biases, we work under knowledge illusions, and the business model still is file first and figure it out later. And once you have FDA approved stamp, and then you claim validated after making 3 batches under most ideal conditions of getting your R&D people to make the 3 batches, same lot of raw materials. So the question really is, how representative is that of commercial manufacturing? Often it is not. And that, so once you are working in a company which says is FDA approved, it's validated, if I don't look, there's no problem, how can you improve? How can you correct your errors? You cannot. So let's imagine the task at hand is to develop a elixir dosage form. What is an elixir? A magical or medicinal potion is one definition, but it is in modern pharmacy a hydroalcoholic solution dosage form for per oral administration. That's a design attribute. To develop an elixir for drug X, what questions should we ask? How sure do we want to be in the answers we get? And what assumptions are we willing to accept? And track those assumptions to make sure that at some later point, those assumptions are not incorrect anymore. What ingredients, process, packaging will ensure a teaspoon of the elixir of drug X over its intended shelf life remain an elixir, that means still have some alcohol by definition, maintain the drug in solution, ensure stability and bioavailability, and prevent contamination and adulteration. Furthermore, we wish to be 95% confident that all we claim in terms of bioequivalence and stability and other aspects. So generally, we will be assuming that bioavailability is self-evident because it's a solution, and all excipients are GRAS. GRAS being an acronym for the phrase generally recognized as safe under certain section of the Food and Drug and Cosmetic Act. One of the most important principles of quality by design is to begin with end in mind. End in mind is a target product profile, which identifies the key quality attributes and the quantities associated with that. Remember, quantities and qualities are objective and subjective, and we'll come back to that later. And then we design a development plan, which is a product development plan, process development plan, and people development plan in one sense. So we need the formulation. So we would, it's a elixir, water, ethanol, propylene glycol is our first choice, 100% by volume to dissolve. The flavor, an essential oil, will include sorbitol for organoleptic considerations, for sweetness, and will buffer if needed. And we'll have drug X, and we'll have some constraints such as keep alcohol to minimum for pediatric considerations or religious considerations. We want to have shelf life of 3 years at certain storage conditions and distribution considerations. And we need to provide weight of evidence and totality of evidence. Weight is where we have uncertainty analysis. Weight, totality is integrative aspects to this to deliver a minimum of 95% confidence interval for key considerations such as shelf life and bioequivalence or bioavailability assessment. And we want to make sure we minimize errors of omission. That means we should do everything we are supposed to do and not miss out on something we were supposed to do. And to ensure low error rate in operations. So operations generally function strive for 6 sigma error rate or less. That is 3.4 defects per million opportunity, which is in terms of confidence interval, a very, very high confidence interval. But 95% confidence actually is only 2 sigma. And so we have to bridge the gap between 2 sigma and 6 sigma in that sense. That's a different mindset, and I'll discuss that in a minute. So the question is, how do we go about executing to develop our development plan? Do we do a 1 factor at a time experiment, or we statistically design experiments considering all the aspects, the whole game plan? We can do mixture experiments, or we can do factorial experiments, or we can do mixture factorial experiments, depending on what the question is and how do we want to answer that question. Now, all of my presentations, I have provided links. The assumption that sorbitol, since the assumption that this is a solution dosage form, so the bioequivalence or bioavailability is self-evident. Our research, my research at FDA on sorbitol shows that a small amount of sorbitol can reduce bioavailability of low permeability drugs from solutions or anything. So that assumption is not really valid. So are you aware of that? And so each link will take you to more additional information for you to think about this. So keep that in mind. So mixture, a factorial mixture are links to some of my own research. Have you considered how different might be your current approach be from the infamous 1937 iconic elixir of sulfanilamide? Are you better able to demonstrate maturity, design, and systems thinking than what happened in 1937? If not, why not? Have you wondered why FDA names that tragedy, which is the elixir incident of 1937, a sulfanilamide disaster? Sulfanilamide was not the root cause of the problem. The root cause was diethylene glycol used as in the elixir. And the only reason why FDA could penalize the company was because there was no ethanol in it. No other reason. So the definitions matter. How we name things matter. Applications to FDA today are called new drug applications. When FDA does not approve a drug substance, it approves a drug product. We still call our excipients inactive ingredients, forgetting about the functionality, forgetting about all the important considerations for that. Certificate analysis of our raw materials is purely based on purity and impurity considerations, not their functionality, not their processability. So you can have supplier to supplier, lot to lot variability, which are not accounted for when you purchase the raw materials, and then you design a manufacturing process fixed in time. You blend for 10 minutes, you granulate for 10 minutes, and you expect to have products which is less variable. You put in variability comes in, fixed process time, variability goes out, and you have all sort of manufacturing problems. So think about this. Are you any better today in your practices of formulation development than 1937? If you review the paper on the FDA website, which the link was there on the previous slide, sulfanilamide disaster, you can hear the voices of a mother who lost her child. You can hear the voice of a physician who prescribed that product, but you don't hear the voices of the formulator or the CEO. When you dig deeper into the literature and into the news media, you hear silence from the formulator. He shot himself, and he shot himself through his heart. On the other hand, a physician CEO doubled down and said he doesn't have to take any responsibility. I do not feel there was any responsibility on our part. When in the literature, toxicity of diethylene glycol was known when this incident occurred. And so who is responsible? So if FDA does not require it, and if compendial monograph does not cite it, then there is a segment of the population, especially the CEO population, which will kill people to make money. That is human reality we are in. Instead of one factor at a time experiment, if we do a mixture design with water, ethanol, and propylene glycol, X1, X2, and X3, and measure solubility, and then we can also measure solubility of drug, of the flavor. We can also relate that to stability. We can also relate that to organoleptic, taste, consumer acceptance, and so forth, is you get a response surface. The response surface was so nonlinear. To make it more discernible, we have taken log solubility. So if you don't plot log solubility, the thing looks quite nonlinear, and the contours are very, very drastic. And so we know where to focus, how the solubility relates to stability. So you'll have multiple such response surfaces, and you can come to an optimal selection. So in this sense, I share with you the link to that paper where I got this slide from. That was my paper research at Cincinnati. But also I would like you to look at my presentation at the FDA Pharmaceutical Inspector training on design of experiment, and especially what we did for metoprolol, sustained release products for SUPAC, scale up and post approval changes. But also I have included in a couple of slides from Amgen and how they use screening studies, screening designs, and then design of experiments to establish a objective risk assessment and control strategy. In 2019, the CPHI expert report that I submitted was Chaos to Continual Improvement, Path to Harmonization. This paper explains systems and design thinking in more in depth, so you may want to review that paper. It's available by just clicking on the link provided. What I wish to do is provide you a context, the current context of a surging revolution, Industry 4.0, amidst a geopolitical chaos and supply chain disruption. What order will emerge from this is unknown, but a source of serious anxiety and in many countries, the risk of famine. What lessons on the PAT to QbD journey to 2020 are still relevant, generally and specifically in the context of this workshop? In summary, I took a deliberate journey to learn from experience, Schrodinger's cat, and my journey from 2015 to 2020, which ended with a Vision 2020 Plus in 2019. And I'm now focused on smart shepherding practices in the chaos we are in currently. So why did I take this journey? And how does it relate to professional development is what I want to talk to you about. Confronting bad eye or breaches in assurance of data integrity when I returned from tobacco sector back to pharma and reconnected with India, provided a passion to empower workers to support their own development. Why did I take this journey? Because I wanted to prevent a clash of cultures as well as empower the people on the front line. India has some excellent talent, but a bad system will beat a good person every time. And we live and work in a socio-technical system. Technical maturity, technical know-how is only one part. It has to go with maturity in terms of social and emotional skills. How to break the pharmaceutical 2 to 3 sigma barrier like Amgen is an article I wrote in 2017. Looking outside in, I had no association with Amgen. While at FDA, they had participated in the workshops that I had held for PAT and Quality by Design to share how they use design of experiments. And that's the reason I asked you to go back and look at my FDA presentation to the Pharmaceutical Inspectorate. But Amgen shared information in the public domain, which I then gathered and tried to understand how did they journey to Six Sigma, that is 3.4 defects per million opportunity, not the Six Sigma methodology, but the low error rate of 3.4 defects per million opportunity. What keeps us from changing? Victims of our own success. Products have strong patent protection. We are isolated from economic cycles. Regulatory system promoted and encouraged inspect and test quality into the products. And there was no sufficient pain to change. So it is so easy in the sector to adopt a mindset, if I don't look, there is no problem. And the FDA inspections happen infrequently and often only for a short period. The likelihood of them detecting any problem is rare. Therefore, why bother? But Amgen made a change. We are here to serve patients. Investigations are important, and they invested into a continuous improvement journey, first by getting a handle on their investigation, corrective actions, and preventive actions. From 2006 to 2012, you'll see the list of steps they took. Going from if I don't look, there is no problem mindset to problem solving to prevention of error reflects on stages of development and maturity of individuals and the corporation. These stages of development align quite nicely with the constructive theory of adult human development and immunity to change that Professor Keegan at Harvard has progressed. According to this theory, constructive means meaning making. Humans are makers of meaning, and how we change, we make meaning is a indicator of our development. So in the case of Amgen, if I don't look, there is no problem. This is a socialized behavior, which Professor Keegan calls third order of consciousness. And problem solving is taking ownership, responsibility to self-author solutions. That's being self-authored as the fourth order of consciousness. And the fifth order of consciousness is to be able to bring the entire corporation, not finger point, to see good in everyone and engage everyone to prevent errors at every stage, at every level of the organization. But to move in this direction, we have to, what Professor Keegan calls, overcome the change prevention system, the feeling system which holds us back, that is the immunity to change, and we have to mature our knowing system. How do I know what I know? So you can see the aspects of a way forward now. On this journey, I learned how to learn from my own experience. Why, despite good intentions, we struggle to fill the gaps between what we know and what we practice, how to empower experiential learning is something I learned. I want to share that with you. What can I do? And what can we do together globally? So there are a series of publications from pharmaceutical new prior knowledge to in and beyond COVID-19 to pharmaceutical team science, which are open access, and I encourage you to get these from the link, and if you're interested, please read those. The key message is doing more is recognizing that our systems are socio-technical systems. Their maturity is our maturity to be suitable and capable to move from don't use, don't tell, and if I don't look, there is no problem, and fail and fix, to be able to predict and prevent harm. Knowledge is experienced or applied information. Why integrating modes of learning, that is education, training, experience is essential. How experience means to feel, and in a professional setting, we typically discourage emotions and we count experience primarily as time on the job. What can we do to change that? Surely we can attend HR emotional intelligence training, but those have never impressed me. And I really get bored with HR training. So what I wanted to do was to perhaps find a direct way, experiencing it myself on a day-to-day basis, on a moment-to-moment basis. And clearly, the 3,000 or so companies that supply drugs to America are distributed all over. They're not Amgens. They can't afford to have this training. So how do we empower each other? Is epistemology, asking each other, how do you know what you know? And what is very important to understand is we are not alone. The Institute of Medicine in 2010 called for redesigning continuing education of healthcare professionals and moving towards continuous professional development, because in their words, the current education system, healthcare education system, has utterly failed. And in 2018, the Institute of Medicine, National Academy of Science, and National Academy of Engineering came out with the proposal of the need for a design thinking systems approach for well-being within education and practice. It's a wonderful paper that you should read, and it points to the need for integrated design and systems thinking, which 20 years ago, the PAT and the Quality by Design initiative at FDA had established. But what we did not do was to focus on experiential learning. How to take a development stance? It is internalizing process validation practices, principles within for internal validation. In testy circumstances especially, I ask, am I seeking to look good? Or just be good? Or do good? And in doing good, I need to feel good too. So I pay attention to intentions. Are they principled? I notice deviations and associated patterns of interrelated feelings, thoughts, and actions. I note these things contemporaneously, and actually I share them on my social media. Internal validation and as process validation makes sense is to make meaning and bridge the asymmetric information, knowledge, and understanding. That is the key. I'm at the moment doing this a trial and error. I hope to soon get into N of 1 nested thought experiments, and I'll report that back to you later. N of 1 clinical trials are the ultimate strategy for individualizing medicine. Now in personalized patient-centric medicine, that's the hottest design of experiment out there. So I hope to come back, share this later soon. The feeling or yearning to be validated by others, external validation, seeking external validation is so familiar to us as teenagers. We want to look good to others. And then we don't want to rock the boat. We want everybody to think we are being good. But those are stages of adult development that does not take responsibility, and the systems thinking does not come from that. When you are self-authored, internally validated, you're not seeking external validation, you have the confidence to do good. And that's when systems thinking begins. But ego comes in there. And once you're able to manage your ego and you're able to bring everyone of your team members with you, and feel good in doing so. You know nobody's perfect, but you're not finger pointing, you're not getting angry, you're feeling good despite their limitations and you're bringing them with you. That's when we are in the flow zone. That's when our feeling space is most optimal in homeostasis and cognition area, as I have shown here. Process information to experience and collect insights by asking how I know what I know, and move your optimal feeling space with you when you move out of your comfort zone. You have to move out of your comfort zone to be self-assured. To go out of your comfort zone is an important stage in our development. You cannot give others assurance of quality with integrity when you're not self-assured. Remember that. Sense making is a distinct process than process understanding. And so keep that in mind. We move from physical domain to information domain to cognitive domain to become situation aware. We go from data information to knowledge. And we have to integrate our emotions. Experience is to feel emotions, and let's remember that. And how we make meaning of words, subjective nouns such as assurance and quality is key. And meaning making and manufacturing maturity, I think, is a column I used to write. So there's an opportunity in diversity, and variation risk has to be managed. That's the key. Heart, brain, guts. Beyond familiar, expanding the feeling space between stimulus and our reaction to take in knowledge-based action with confidence. Care is research, knowledge is power, but wisdom is in practice. I wrote an article on 4 life science lessons from the PAT to QbD journey recently. Please take a look. From known unknowns to unknown unknowns, we make sure that our intentions are principled. We expand our awareness. We are vigilant to make good observations, and we pay attention to knowledge. That's the totality of evidence, and depending on where you are in a complicated system or complex system, the weight of evidence shifts. The 1962 Kaifa-Harris Amendment requires professionals to have scientific training and experience such that they can assess effectiveness fairly and responsibly. It provides the legal basis, the principled basis to think about interpersonal, inter and intrapersonal and cognitive dimensions, and bring that into our day-to-day practices. The discussion so far has been based on experience, but it is also grounded in theory and scientific empirical evidence from, say, for example, Professor Keegan's work at Harvard. A maturity model for smart I or smart me emerges from that. Why the I or me confusion? I am the subject and me an object of a sentence. So you choose whether you want to be a subject or an object. Select smart I or smart me. Human professionals will be competing with artificial intelligence. Who will win, and whether by the time we reach a industry 4.0 adaptive plant, whether we will need humans in manufacturing or not is an open question. Currently, most of the companies are struggling to get out of the digital silos and to achieve the connected plant. Few have progressed beyond that, but the revolution is marching on. So either you are part of the system as a contributor, or the system will be controlling you. So are you ready to work in a smart factory? Apart from cyber attacks, systems or general attacks, systems gradually degrade, and their degradation can be detected in error rates and seek and settle time. Self-monitoring and reporting technology generates a smart message to alert of an imminent failure or crash. Observations and warning letters are also smart messages. FDA is moving towards maturity of quality system and quality metrics. Smart shepherding practices is what you need for smart pharmaceuticals 2021 to 2030. Thank you for listening. Why emphasis on epistemology? How did the PAT QbD journey begin? A 360-degree view of quality by design are some of more detailed presentations available on my YouTube channel. So if you're interested, please go review those, subscribe to the channel, and connect with me on LinkedIn for continuing discussion on smart shepherding practices. Thank you. I look forward to answering some questions. Remember to take a development stance, learn from experience, and share knowledge. I have included some additional slides since this is a workshop, a challenge to you, a legacy challenge in the experience economy. So take a look and see how these might help stimulate your thought. Welcome to the experience economy. Do you know we live in a post-truth world? And trust in expertise is perhaps the lowest I have ever seen. The current systems evolved after the Western Enlightenment. Where are we today? Formulation development from art to science. How did the FDA PAT initiative challenge this paradigm? Are you better than market standards? Do you know what is continuous improvement and what is needed in how we set specifications to improve continually? In very simple terms, by design means by intent, to plan something for a specific role or purpose. Do you know what are the essential elements to write a quality target product profile? Remember, assurance in and of itself is a critical quality attribute. Placebo and nocebo effects are expectancy effects that patients feel. They're not just psychosomatic, they have physiological implications. Beware of regulatory defaults. Stop saying, if I don't look, there is no problem. Epistemology necessary to break the Procrustean bed. 4+ order of consciousness is at minimum our suitability and capability. That's what qualifies us.